Yeah. Okay, are there any questions before we get started? Let me share my screen. Can somebody confirm you can hear me, please?
I can hear you.
Thank you. All right, today is February 7th, and we're going to be discussing Lab 6, disc diffusion and antibiotics. Lab 6, disc diffusion, evaluation of disinfectants and antiseptics, and antibiotic effectiveness. As is normal, there is some reading to do before beginning this lab, and the reading comes from your textbook. There are some video clips today, and I don't remember, is there, come on, stop that. It looks like there's a virtual lab as well. All right, the learning objectives, be able, one, to identify the type of chemical agent and the active ingredients used in various disinfectants and or antiseptics. Two, evaluate the effectiveness of disinfectants and or antiseptics using the disc diffusion method. And we'll talk about that in this lab, the disc diffusion method. Three, determine the mode of action of various antibiotics. Four, evaluate the effectiveness of various antibiotics using the disc diffusion method. Five, list variables that influence the antibiotic disc diffusion susceptibility testing. And six, be able to define the term sterilization, disinfection, disinfectant, antiseptic, bactericidal, bacteriostatic, antibiotic, spread plate, and zone of inhibition. Any questions about learning objectives or what we're covering? All right, in this lab, we're largely going to be talking about the disc diffusion method. It does have other names. It's also called the Kirby-Bauer method after the two scientists who, I don't know, discovered it. It is a way to determine the susceptibility of bacteria to different antimicrobial agents. The Kirby-Bauer method is almost always used when testing for antibiotic resistance or sensitivity. It may also be used in other tests, like testing for a chemical to see if it's antimicrobial, like various disinfectants and antiseptics. It is not as good as the antibiotic testing, but it is good. Introduction. Sterilization eliminates viable organisms. So something that is sterilized has no viable organisms in it, and this includes endospores. In the lab, how we sterilize things, if you were to come into a normal microbiology lab, is we put things in an autoclave. Disinfection refers to the destruction of vegetative cells, not necessarily destroying endospores. A disinfectant is a chemical agent applied to an inanimate object. So when you're using bleach on your toilet, the bleach is a disinfectant. It does remove most, well, all vegetative pathogens and vegetative cells, but bleach does not kill all endospores. Antiseptics, on the other hand, is a chemical agent applied to living tissue. So if somebody were to put bleach on their cat who got cut and infected, and they use the bleach, it would be an antiseptic. So the same chemical can be a disinfectant, and an antiseptic, depending on how it is used. Usually, antiseptics are not as harsh and irritating to the skin as a disinfectant is, meaning bleach is pretty irritating to the skin, and so it's not usually used. However, one of the older treatments, and I don't know if it's still used today, but it was an effective treatment for stopping necrotizing fasciitis, was to wrap the affected tissue in some gauze that had been treated with bleach. And that was at least one of the effective early treatments for stopping necrotizing fasciitis. And obviously, they'd give antibiotics as well. To evaluate the effectiveness of chemical agents, the surface of a nutrient agar plate is inoculated with a particular bacterium. Usually you get the bacteria to grow all over the plate, and we call that a lawn of bacteria. Paper discs saturated with different chemical agents are then applied to the surface of the agar. The chemical agent diffuses into the agar, establishing a concentration around the disc. You then incubate the plate overnight and look at the bacterial growth pattern around the disc. If a zone of inhibition appears, this is an area of no bacterial growth, the chemical is assumed to be at least effective for that particular bacterium. Conversely, if no zone of inhibition appears, the chemical is assumed to be totally ineffective. Now, this is a gross way of looking at it, because to be totally effective, and let me see, if to be sensitive to something, you need to do more than just see a zone of inhibition. The zone of inhibition has to be a certain size. And then to be resistant, you don't have to have no zone of inhibition. You could have a zone of inhibition below a certain size. And we'll talk more about that in this lab. So this table is telling you the different chemical agents, their general action, their preferred use, like ethanol and isopropanol, are used as an antiseptic. They are not very good antiseptics, so we call them degerming agents, meaning when we apply either ethanol, isopropanol to the skin, we do not sterilize the skin. All you do is you reduce the number of microbes present in the place where you applied the alcohol. Formaldehyde, formalin, glutaraldehyde are disinfectants, and these are good disinfectants in that they actually kill endospores. There are not too many disinfectants that kill endospores. Like halogen chlorine bleach is a disinfectant, but it does not kill all endospores. Heavy metals, mercury and silver, their use has come back in the health setting because of the resistance of bacteria to chemical agents, particularly antibiotics. And so we're reusing the heavy metals, and sometimes gauze will be treated with heavy metal to prevent bacteria from growing like in an infection. Peroxygens, like hydrogen peroxide, is a very good antiseptic and is one that will kill endospores. Phenolics and other mostly disinfectants. Surface active agents, disinfectants, soaps, things for washing your hands. The zone of inhibition size depends only partially upon the sensitivity of the organism to the disinfectant or the antiseptic. There are various other factors that affect the zone size, including the concentration of the chemical. Generally, the more concentrated the chemical, the larger the zone of inhibition. The larger and the molecular weight can affect the size of the zone of inhibition. Generally, the larger the molecular weight of the chemical agent, the smaller the size of the zone of inhibition. And then the density of the bacterial inoculum can also affect the size of the zone of inhibition. Generally, if the bacteria are more and more dense, you'll have a smaller zone of inhibition. Evaluating antibiotic effectiveness. Antibiotics are categorized as bactericidal if they kill bacteria, and bacteriostatic if they only inhibit the growth of the bacteria. If you inhibit the growth of the bacteria by a bacteriostatic agent, and then you remove the bacteriostatic agent, the bacteria may regrow. There are antibiotics which are both bactericidal, meaning they kill bacteria, and there are some antibiotics that are bacteriostatic. When you apply a bacteriostatic antibiotic, you're relying on the host immune system to assist in the elimination of the infection, because the antibiotic that's bacteriostatic simply stops the growth of the bacteria. It does not remove them. Any questions about any of that? All right. The Kirby-Bauer disk diffusion test, which is the thing I was talking about up here called the disk diffusion method, is a test that is frequently used to test for antibiotic testing, meaning to test if the bacteria are sensitive or resistant to the antibiotic. It may be used on different chemical agents like antiseptics and disinfectants, but it is very good on testing for antibiotic resistance or sensitivity. The results of such testing help physicians choose which antibiotic to use when treating a sick patient. Bacteria are isolated, grown in an agar plate, and then filter discs impregnated with the antibiotic is applied to the surface of the agar. The antibiotic will then diffuse away from the disc, creating a zone of antibiotic, and the concentration gradient around the disc can result in a zone of inhibition where the antibiotic concentration is high enough to inhibit bacterial growth. Now, in this zone of inhibition, it may kill the bacteria. Let me blow this up. Or it may just inhibit the bacteria's growth. Either way, you get a zone of inhibition. So we don't know if that's from a bacteriostatic agent or a biocidal agent. You would think that the size of the zone of inhibition would be a measure of the sensitivity of the bacteria. Like this, you would say this bacteria is sensitive to this antibiotic, but fairly resistant to that antibiotic. That is not the case, though, because there are several factors coming into play to determine the size of the zone of inhibition. First, it is true that the sensitivity or the resistance of the bacteria will determine the size of the zone of inhibition. But let's say you have a very small antibiotic. It's going to diffuse very rapidly and further in the agar than a very large antibiotic. And then there's a few other factors that come into play, such as the growth rate of the bacterium. Table 6.2 lists the procedural protocols required to standardize the variables that will affect the size of the zone of inhibition when performing the Kirby-Bauer disk diffusion method. So you first, let me make sure I got that small enough to read. Move it over just a little. There we go. First, you inoculate a nutrient broth using several bacteria colonies that have been growing on a non-selective media, but they've been growing for no more than 24 hours old. You then adjust the broth so the turbidity is 0.5 McFarland standard units. This is approximately 1 × 10⁵ bacteria cells per mil. You then inoculate room temperature Mueller-Hinton agar plates. So we always use Mueller-Hinton agar plates, and the different types of media can affect the size of the zone of inhibition. The Mueller-Hinton plates are always between pH 7.2 and 7.4. So we pH them to make sure they're between pH 7.2 and 7.4. Furthermore, you put the agar at a certain depth in the plate, always using 4 millimeters of agar. The different depth will affect the diffusion of the antibiotic through the agar. And that is, if there's more agar, higher depth, then the antibiotic will diffuse less than if there's less depth. So we always use 4 millimeters. And then furthermore, we always use a plate that's 150 millimeters across, meaning the diameter is 150 millimeters. And then we apply the bacteria on we're using a non-toxic swab using the spread plate technique, which we'll discuss in this lab. And you swab the plate everywhere to obtain confluent growth on the plate. You then let the media containing the cells soak in for about 5 minutes, and then you place discs at least 24 millimeters apart, no more than 12 discs on a plate, and you add the antibiotic disc between 5 minutes after to 15 minutes after adding the bacteria to the plate. So you let the bacteria sit for 5 minutes, and then you start applying the disc. You make sure the last disc goes on no later than 15 minutes after putting the bacteria on the plate. You then incubate the plates upside down in a non-CO2 incubator for 16 to 18 hours, growing them at 35 degrees Celsius. And you put the plates in the incubator no more than 15 minutes after adding the antibiotic disc. So these are all the standard protocols we use to ensure that only the bacteria will affect the size of the zone of inhibition. And this way, if you're using the same antibiotic, no matter where you are in the world, whoever doing the test, they should get similar results, meaning they should get a similar zone of inhibition for a particular antibiotic for that same bacteria. So if we're looking at E. coli and you're applying streptomycin, you should get a similar antibiotic from somebody else running the same test someplace else in the world. After incubation, the inhibition zone sizes are then measured using a ruler in their diameter. You measure using millimeters. The bacteria is considered to be sensitive S or intermediate I or resistant to the particular antibiotic based on the test and on a table. For example, here is the zone of inhibition for, I believe it's Staphylococcus aureus. The antibiotic here is penicillin, that's penicillin G. You measure the size of the zone of inhibition. This looks like it's about 20 millimeters, the diameter, and usually you measure that twice, meaning you measure it this way and then that way. And if it's different, you take the average. But this is 20. And then you compare it to a table. The table says that if the size is 20 millimeters or less, the zone of inhibition, then this bacteria is resistant to this antibiotic. And if the size of the zone of inhibition is between 21 to 28 millimeters, the bacteria is intermediate to this antibiotic. And if the size of the zone of inhibition is equal to or greater than 29 millimeters, then we say the bacteria, this one here, is sensitive to this antibiotic. Any question about that? So to determine the sensitivity or the resistance, you have to grow up the bacteria on plates having the antibiotic, and you have to apply the bacteria using this standard protocol and applying the antibiotics using this standard protocol. Grow it between 16 to 18 hours at 35 degrees Celsius. Measure the diameter of the zone of inhibition, and then compare it to a table. This table comes from the NCCLS, the National Committee for Clinical Laboratory Studies, makes the table. And I'm just going to call it the NCCLS standard table. Each antibiotic will have a different size for its zone of inhibition if it's resistant to that antibiotic and if it's sensitive. Like streptomycin will have a different resistance size for penicillin G and a different sensitivity than penicillin G. And each bacteria can have a different diameter that's resistant in sensitivity for the same antibiotic. Any question about any of that? All right, so know the terms of this lab. You're not going to be physically performing any of these activities, but you will be responsible for how to interpret the effectiveness of antibiotics using the Kirby disk diffusion test. In procedure 1, we have a little video for you to watch on preparing a spread plate. So watch that video. And then review the steps for using antibiotic susceptible technique when preparing a spread plate or a bacterial lawn of growth. Let me go ahead and demonstrate that. Pretend this is a cotton swab. Let me see, I have an open container here someplace. This is my tube of containing bacteria, and here is my plate. What you do is you take your cotton swab and it's sterile, put it in the media containing the bacteria, and then before you take the cotton swab out, I don't know if you can see this, but you touch it to the side of the tube to get the worst of the drop of media off the cotton swab. So you don't want it dripping and you don't want it totally full of liquid. You have to keep it sterile. Then you take your plate and then you inoculate everywhere on the plate. I don't know if you can see this, but I'm trying to inoculate everywhere on the plate with the cotton swab. Then you rotate the cotton swab a fourth of a turn. You rotate the plate a fourth of a turn. And then once again, you swab everywhere on the plate, putting the bacteria everywhere on the plate. I'm not doing a very good job, but you're getting the idea. Then you rotate the cotton swab a fourth of a turn. You rotate the plate a fourth of a turn. And you swab the plate everywhere again. You do that at least 3 times. Then you take the cotton swab and you move around the edge of the plate, which is hard to show you, but because I don't really have a plate here. The reason is because when you're swabbing like this, sometimes you won't get the edge. And so you take your cotton swab to get bacteria all along the edge. This will put the bacteria everywhere on the plate, and then you will grow it as a lawn of bacteria. Bacteria will grow everywhere. Sort of like grass is everywhere in your lawn. If it's a good lawn of grass, you want the bacteria to grow everywhere. Do not do, I once had a student who simply did do, 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 and then she had these humongous holes with no bacteria on the plate. And she said, well, how can I measure the size of the zone of inhibition? And I said, when you swab the plate, you didn't swab it everywhere. And so you don't have bacteria everywhere. You have to have bacteria growing everywhere in order to measure the diameter of the plate. And I told her she had to either repeat the experiment or she could use her lab partner's data. Because hers was not any good. You then allow the liquid containing the bacteria to be absorbed by the media for 5 minutes, and then you put the disc containing the antibiotics on the plate. And then you take the plate and put it upside down. So I guess that's right side up in this case. So we put it upside down and then stick it in the incubator upside down. Incubating it overnight at 35 degrees C. And you do that for, incubation is for 16 to 18 hours at 35 degrees C. All right, in procedure 2, you're going to be evaluating the antibiotic effectiveness. We have a video clip on preparing a culture for Kirby-Bauer disc diffusion method. And then we have a video clip on the interpretation of the results for the Kirby-Bauer disc diffusion method. Let me go to the virtual lab now. And it says to use the new link. So I probably got that new link on the worksheet. You might have it on the lab module, but I don't. I don't have it here. So let me go to your. The old link didn't work anymore. So that's why I'm trying to use the new link. Come on. There we go. All right. Depending on how fast your internet connection is, you may have to wait for this page to load. Click Start. And this shows you the Kirby-Bauer disc diffusion method test. And it's already been incubated. The different antibiotics are here. TM stands for, I don't know that antibiotic, tropramycin. So let's go S. That one is probably streptomycin. That one I know of. And then you take the little ruler to measure the diameter. And that looks like the diameter is about 18 millimeters for streptomycin. Come on over here. And if it's 18 is greater than 15, you select that. If it's green, it means you got the right answer. For TE, it's probably tetracycline. Yep. So here that looks like about 18 too. I'm going to take the wrong answer and say 19. If you get the wrong answer, to go in X. Wrong answer, X. So it's actually 18 and it's green. All right. So this is how you do the virtual lab. And then you can click the menu. There are actually 3 plates here. Restart, and it switches the plate. So this is Pseudomonas aeruginosa and the different antibiotics here. And then you got another table there. And you can go and do it again. And there's Staphylococcus aureus. And boy, there's only one that looks like it's having a zone of inhibition. Is that vancomycin? This must be what do you call the MRSA, multi-drug resistant Staphylococcus aureus, because the only one that's working is vancomycin. Let me get that out of the way here. Anyways, that's how you do it. You don't need to look at the description, but you can. And you don't need to do the steps, but you can. And you don't need to look at the credits. Anyways, that's how to do the virtual lab. You'll need to do the virtual lab to get some of the answers in your worksheet for this lab. Oh, I guess I'll leave that there just in case. Let's go to the worksheet, although I'm not there yet, but there he is for question 11. You'll need to get that from the virtual lab. And this tells you a little bit how to use the virtual lab, but I just showed you. All right, 2, using the standards required for the Kirby-Bauer susceptibility testing, an E. coli culture is prepared using a spread plate technique. Antibiotic discs are placed on the surface of the agar. The plate's incubated, and below are the results along with the NCCLS zone standards used for interpretation. And this I have in the worksheet, so let's go to the worksheet now. Come on. So this is the antibiotic disc, and the antibiotic here is ampicillin. This is the size of the zone of inhibition. We're measuring the size of the zone of inhibition by calipers. How you use a caliper is you do not read from here to here, which would be about 9.5. Let me blow that up. I can't blow it up here. Let me go that way. 9 to 9.5 millimeters if you were to read it there. You do not read a caliper that way because if you notice, the units, the measuring does not start here. It starts there. And so you read the caliper from 0 to 0. And so we read it as here. That's really close to 15. It's a hair less than 15. It's more than 14.5. So I'm just going to say 15 for ampicillin. I'll do the first one for you. So the size of the zone of inhibition is 15. And then the interpretation is either S, I, or R. You come up to the table here. Remember, we're using E. coli, and then ampicillin, cephalosporin, gentamicin, and tetracycline. So E. coli, and the table is here. Ampicillin is here. We said 15 was the size of the zone of inhibition. And so 15 is going to be I, intermediate, right here. And so you put an I in right here. And then you measure the distance in millimeters for cephalothalin, CF, I'll word it that way. I have trouble saying that one. Right here, measure it from here to here, compare it to the table, and then state your interpretation here. So you need to put in the size of the zone of inhibition here, as well as the interpretation there. Any question about any of that? All right, question 3, list the 5 variables that must be standardized when performing the antibiotic susceptibility test using the Kirby-Bauer disk diffusion method. 4, the size of the zone of inhibition often varies for 2 different species of bacteria when challenged with the same antibiotic. What is a possible explanation for these differences? 5, what would happen if we didn't use aseptic technique to put antibiotic disc on the plate? Mention what will happen to our test results. 6, what would happen if we increased or decreased the concentration of the antibiotic in the disc? Be specific in your answer. You also need to state both what will happen if you increase the concentration of the antibiotic, as well as what will happen if you decrease the concentration of the antibiotic. If you only answer for one, you'll only get partial credit here. And like I said, be specific in your answer. 7, antibiotics can be bactericidal, that is, they kill the microbe, or they can be bacteriostatic, they inhibit the microbial growth, but do not kill the bacteria. When observing the zone of inhibition around the disc, how would you determine whether the antibiotic causing that zone was bactericidal or bacteriostatic? I'm going to warn you that this is a thinking question. You will not be able to get the answer directly simply by looking it up in the lab module. You have to think about the answer. Students often get this question wrong. I will say about half of you will get this wrong the first time. And some terms I've had every student get this wrong the first time. Like I said, this is a difficult question. It does require a lot of thinking. But generally, after you've thought of it, and maybe after the second grading of the lab, the majority of the students do get this question correct. And if you don't get it correct the second time, I will tell you how to correctly answer that question. All right. When performing antibiotic sensitivity testing, it's important that the test conditions are kept very standardized, meaning we use the same kind of media, Mueller-Hinton median. The agar depth is always the same. The age of the culture is always the same. The pH of the culture is always the same. Why? So discuss why. 9, are there any other substances besides antibiotics that we could evaluate using this method, meaning the Kirby-Bauer disk diffusion method? And you should state at least one use, like some thing that you're testing using the Kirby-Bauer disk diffusion method that is different than testing an antibiotic. 10, does the presence of a zone of inhibition necessarily mean that the bacterium being tested is sensitive to the antibiotic? Explain your answer. This is a thinking question, but this one you can get by looking it up in the lab module. 11. From the size of the zone of inhibition of chloramphenicol on E. coli in the virtual lab, is the bacteria sensitive, intermediate, or resistant to the antibiotic? For chloramphenicol on Pseudomonas aeruginosa, is the bacteria sensitive, intermediate, or resistant to the antibiotic? Any question about any of that? All right, if not, that's it. I will be here. Let me continue recording. I'll be here to help you in the lab to answer any questions. And I will be here at least until 8 o'clock, assuming that students do not log out. But once the last student is logged out, I too will log out. Any questions? All right.
I was just asking, what is the quiz, like what chapters is the quiz on for today?
I'm sorry?
For the quiz,
The quiz
what,
today?
yeah, what are the, like, what chapters are the questions, like, I guess?
Okay, let me start sharing my screen because I'll show that. You just need to look on the syllabus. It'll have it in the schedule. So the quiz for today is going to cover labs 3 and 4 and chapter 3 and the end of chapter 2.
Okay, thank you.
Okay. And I believe this is one of the quizzes that I actually moved for you guys because we have a different lecture schedule. All right.